The cytoskeleton, which, as you may suspect, is going to have a lot to do with the ability of some cells to be able to move around. Okay, so we kind of combine these two topics kind of together. Okay, we can sort of divide the cytoskeleton into these three major types of elements. So just like our skeletal system, our musculoskeletal system has muscles and bones and joints and connective tissue and cartilage and all that kind of stuff. The cytoskeleton also has several different types of components, and the three major ones, I think we've looked at this picture earlier in the course for immunofluorescence microscopy, but it did turn out to be showing you the three major elements of the cytoskeleton. So the red ones here are actin filaments. These are the smallest of the three. They're sometimes also referred to as microfilaments. Microtubules are the green ones in the middle. That's where we're going to actually start. These are the largest of the three types of elements. And in between, intermediate, between them, in size at least, are the intermediate filaments, although we're going to hold them to the last. So we're going to do microtubules, then actin, then intermediate filaments. Okay, so obviously we can visualize them quite nicely using immunofluorescence microscopy. So we can study them in terms of, if we throw specific molecules or drugs at the cell, we can see if they have an effect on the organization of the cytoskeleton and things like that. The next couple of slides are taken from your textbook. They're just sort of basic summaries of some of the characteristics of these three types of cytoskeletal elements. So I'm not going to read these to you. We'll cover much of this material as we go through the topic, but the slides are here, again, sort of as a summary kind of idea. Sort of, again, some summarized information here in sort of table form for the three elements in terms of basic structure, diameter, monomers, polarity, things like that. So again, you can have that as a summary kind of at the end. Okay, so we're going to start again with the largest elements, the microtubules. And you're probably at least somewhat familiar with these from Bio 110 if you studied mitosis and meiosis. One of the major functions of microtubules is to move chromosomes around during cell division. So we'll come back to them as well when we get to that topic a little bit later in the course, back out in December. So if we kind of start at the smallest level, microtubules are made up of proteins called tubulin. And it turns out that there are at least three different kinds of tubulin, and they're given Greek letters to designate them. So there's alpha tubulin, beta tubulin, and gamma tubulin. And the basic structure, or at least the beginnings of the basic structure for microtubules, is composed of the first two. So the alpha and beta monomers get together to form a heterodimer, as you see at the start up here. And so alpha and beta get together and dimerize. When they sort of hook up together, they sort of kind of look like a dumbbell-shaped protein with one alpha and one beta subunit together. So that's the term. That's where we get heterodimer from. So that essentially is going to be the subunit to make a microtubule, are these alpha-beta heterodimers. And they are going to start polymerizing into a long chain that's called a protofilament. Okay, so we have monomers, dimers. The dimers are going to polymerize into these protofilaments. And notice how they're doing that in this figure. They're always going beta, alpha, beta, alpha, beta, alpha, beta, alpha, beta, alpha, beta, alpha. You get the idea. These could be hundreds or thousands sometimes of dimers long. But they always line up in that specific way. Okay, so it's never two beta ends touching, and the two alpha ends, it's always beta, alpha, beta, alpha, beta, alpha. Okay, so there is then inherent polarity to these protofilaments if they always line up that way. Right? So we have one end of the protofilament has the beta subunit there, the other end has the alpha subunit. Okay, so there is then inherent polarity, again using that in the broadest sense, to protofilaments and therefore to microtubules as a whole. Okay, just like we have the N terminus of the C terminus of a protein, or the 5' end, the 3' end of DNA, we have polarity here. Now, we've come across a number of instances where they've named things really, really badly. Here's another one. So instead of just calling them the alpha end and the beta end, which I think would be really smart and reasonable, of course we can't do that. Right? We've come across other examples of that. So instead of just calling it the beta end and the alpha end, which I think would be a good idea, somebody came up with a really bad idea of calling it the beta end the plus end, and the alpha end the minus end. Terrifically bad idea, especially in a course like this. Because if you hear plus end and minus end, what is your automatic thought? Charges. But it has absolutely nothing to do with charge. So write that down. Okay? Plus end, minus end, nothing to do with charge, just a really poor naming system. So that's the actual designation for this polarity, plus n, minus n. But as usual, I don't get the name stuff, so we have to live with it. Okay, but please realize the plus and the minus have absolutely nothing to do with charge. That is not positively charged, that is not negatively charged. It's just a really, really bad name. Okay, but the really important thing is microtubules, starting with the protofilaments, have polarity. Okay, and this plus n, minus n will make a really big difference in terms of how these things work and how they're organized in the cell. So despite the bad name, the polarity idea is really important here. Okay, so we go from monomers to dimers, dimers to protofilaments, protofilaments to the microtubule itself. So if you look around the the perimeter, the circumference of a microtubule, how many protofilaments do we see? We'll just start there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Lucky 13. So 13 protofilaments make a microtubule. So these are very long but hollow structures, cylinders basically, tubes. So there seemingly is nothing inside them, so these are just hollow, really long tubes or cylinders. And I always like that they give us micrographs, so again, we're not making this stuff up. Okay, so here's a cross section through a microtubule, here's a longitudinal section through one. And all the protofilaments, as you can see here, all have the same polarity. Okay? So all the beta ends up here, all the alpha ends down there. So like a lot of complex structures in the cell, they're built up of smaller, individual monomers or subunits is probably the better word. Okay, so we have these heterodimers, protofilaments, 13 protofilaments make a microtubule. And that always seems to be so. Okay, so that seems to be the standard way of creating. You don't have some with 12 and some with 15. It seems to always be 13 for whatever reason. Okay. So that's sort of the basic structure of these microtubules. Does that kind of make sense? Okay. Okay.
When does the other one kick in?
Eventually. Shortly, actually. Okay, so we have all these microtubules around, so Yeah, if we go back to that original picture, right, there are a lot of these microtubules around. And so each of these green lines is a microtubule. And it turns out that microtubules are the only one of the three that we're going to look at that require what's called an organizing center. So we call it a microtubule organizing center, an MTOC. And there are going to be a couple of types in the cell, but the major one for the cytoplasmic microtubules is the centrosome, which is shown here. Hopefully you remember that from at least Bio 110 in terms of this is one of the things we look at, say, in prophase of mitosis, right? The centrosome copies itself, and then the two copies move to opposite ends of the pole, or opposite poles of the cell, opposite ends of the cell. And it turns out that they do that because out from the centrosome grows all of these microtubules, as we'll see in a second. So the centrosome, in animal cells at least, is sort of this kind of gelatinous stuff, this sort of green goop here. And if you'll look closely, you have these little rings, this little sort of orangey to me here, these little orange rings, and the orange rings are the third type of tubulin. And we think that those little rings of gamma tubulin are sort of the starting points for the microtubule itself to sort of organize there and start kind of growing out from these little rings of gamma tubulin that are embedded in this sort of, like I said, sort of gelatinous stuff of the centrosome. And then in animal cells, but not plant cells, only animal cells, if you look inside, they have a pair of what are called centrioles. So you have this sort of gelatinous matrix. You have all these little rings of gamma tubulin. I'll just draw a few in. And then inside, you have this pair of, I'm just going to draw them kind of simply here, what are called centrioles. So they're in plant cell, or I'm sorry, no, they're in animal cell centrosomes, but not in plant cell centrosomes. So that's kind of weird. I'll come back to this side in a second. Let's take a look. So if we look at the centrioles, they're a little bit, not too complicated, but they're more complicated than we're kind of letting on here. So this is really what the centriole looks like. And there are always two, and they're always organized sort of perpendicular to each other. You really don't know why, so one sort of points up and down and one points this way. But they're not just single tubes, as I've kind of badly drawn over here. They're a little bit more complicated than that, as we see here. They're actually, we start at the top, 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 triplets of microtubules. So there are actually 27 microtubules here that are all sort of fused together in sort of triplets, and they're sort of held together with other proteins. So there's one of these, again, sort of pointing up and down. There's another one pointing left to right, I guess. And that's what a centriole is. What it does is sort of anybody's guess, because animals have them, but plants don't. The plant centrosomes work just fine without them. So we still don't really know what these things are doing in the middle of this centrosome thing. But they are there. That's kind of what they look like. Don't really know what they're doing. Because we really think that it's the rings of gamma tubulin that are the real sites of sort of construction of the microtubules. So what these are really doing, we're not really sure. But this structure we're going to see in another thing called a little bit later called a basal body, which looks very much like this centriole. So there is going to be a connection a little bit later with another kind of MTOC, another kind of organizing center called the basal body. So we'll see that eventually. So again, an electron micrograph of these things. And then this is kind of a neat picture because it shows the centrosome there with all these microtubules coming out from it. So if you had the ability somehow to kind of trace all of these microtubules, they should theoretically all trace back to a single point, and that single point is the centrosome. It would be almost impossible to really do that here, but you should be able to do that. You should be able to trace all these microtubules back to that single centrosome in the cell. So it's vital to the cell that it has a centrosome. Okay? And we know that very clearly because if you kind of zap this with a laser, all the microtubules disappear and they never come back. Okay? So you absolutely need, the cell absolutely needs a centrosome to construct all of those microtubules. It's an absolute requirement. And the way they're organized takes us back to that polarity stuff that we were just talking about. It always seems that the minus end, the alpha end, is stuck, basically, in the centrosome. And then the other end, that's out in the cytoplasm somewhere, is the plus end. Okay? So that polarity comes into play here as these microtubules are sort of built. So I'm just going to draw them simply as just straight lines. So all the microtubules that are coming out of the centrosome are all organized like that. It's always the minus end at the centrosome, the plus end out in the cytoplasm somewhere doing what it's doing. And so again, that's why this polarity idea is really important, because that's how it's always sort of organized. And that's what they're trying to show us here. So it's always the minus end in the centrosome, plus end out in this cytoplasm. So far, so good? Making sense? Okay. So this is showing one sort of model about how these microtubules may form. So again, we have sort of this population of dimers that start polymerizing into the protofilaments, and then protofilaments sort of start hooking up with each other, and then when they get to that magic number 13, it sort of starts wrapping around into the microtubule, and there's your long, hollow cylinder. It does appear, there's lots of good evidence to suggest that all of this happens spontaneously. So if you have a test tube and just has the alpha-beta dimers in it, no other proteins in it, under the right conditions, they'll spontaneously form microtubules. So all of this seems to happen on its own without, other proteins kind of helping to, construct these long, hollow tubes. So they kind of construct themselves in a sense. Now, one huge reality, this isn't a very good picture of it, of microtubules is that, this is the one I wanted, are that they're very dynamic. So they don't just grow to a specific length and then that's all there is to the story. Most microtubules are sort of constantly getting longer and shorter. They're very dynamic structures. And once we talk more about what they do for the cell, we'll see why that's probably a really good characteristic. Okay, but some of them are lengthening, some of them are shortening, some of them are, staying roughly the same size. But generally speaking, there's a lot of dynamic characteristic to the population of microtubules in most cells. So they are very dynamic structures. So the question is, how does that kind of work? How do they elongate? How do they shorten? And this is kind of a model for how that sort of happens. So once a microtubule sort of starts forming, one thing that's been shown pretty clearly is that you can add more tubulin dimers to it, and therefore it should theoretically get longer, right? So that's kind of what they're showing here. And what's also been shown real clearly is that that takes energy. Okay? So if you want to add tubulin dimers to a microtubule, make it longer, that's going to cost you energy. So to the beta subunit of each dimer is attached GTP. So adding each monomer, or not monomer, each dimer to the microtubule is going to eventually cost you a GTP, cost you an energy molecule. And so these very dynamic microtubules are costing the cell a good amount of energy because, again, a lot of them are elongating quite often. So every single monomer, keep saying monomer, every single dimer costs you a GTP eventually, but not initially. So you need the GTP attached in order for new dimers to add to the plus end. So they start adding to the end that's out in the cytoplasm. Okay, so if you're elongating, say this one, then you're adding new dimers to the plus end, and then it grows out that way. And in order for them to add on, they have to have a GTP attached. Okay. Now the other kind of interesting thing about molecules is that the dimers that we're adding to the plus end start moving down the length of the microtubule. So I like this picture a lot. Okay, so if we color code that little set of dimers, they add on to the plus end, you could literally watch them over time move down the length of the microtubule, and eventually they fall off the minus end down at the centrosome. It's a process called treadmilling. So these are incredibly dynamic structures because even when you start adding things on, they start moving down the length of the microtubule. So even if a microtubule looks like it's staying the same size, now with this treadmilling idea, what do we now know? They're always losing stuff at the minus end. If we want to keep it the same size, what has to happen at the plus end? Yeah. So if they're losing, 10 a second, then if we want to keep it the same size, then we have to keep adding 10 every second. Right? So if we want it to get larger, actually, what has to happen? Right, we have to add them at the plus end faster than they're being lost at the minus end. And then if the thing is shrinking, what's probably happening? Yeah, you're either not adding any more, or you're adding them at a rate slower than the rate at which they're leaving at the minus end. So these things really, really are dynamic little structures. So how does this energy come into play? Well, the GTP is sort of your admission fee, basically, to add on to the microtubule. And then as this treadmills down, as the dimers move down the length of the microtubule, at some point, after they add, but before they leave, before they fall off, that GTP is hydrolyzed. Okay, so you kind of have the check in your hand as you add on, and sometimes before you fall off, the check gets cashed, if you want that kind of analogy. So they've tried to color code that for us here, so the GTP is the orange, and then when it gets hydrolyzed, they color it in red. So you can see at some point after they add, but before it falls off, it gets hydrolyzed. So it's kind of a neat system. So like I said, even if the thing looks like it's staying the same size, it's actually still a very dynamic process because of this treadmilling idea. Does that kind of make sense? We're going to see a very similar situation when we look at the actin filaments when we get to those. Okay, so these things are very dynamic, like I said. Some are increasing in size, some are decreasing, some look like they're staying the same size, but now we know that even that's a dynamic situation. Okay, so these very dynamic structures made up of all these alpha-beta heterodimers and this polarity and the microtubule organizing center. What the heck are all these things doing for the cell? They seem pretty important. And it turns out that they really are quite vital to all cells. Now, some cell types will do some extra things, but even if we start at the bottom here, okay, most of our cells at some point divide. So all cells are going to need microtubules in order to do mitosis. Right? So we'll focus in on that particular function when we get to that topic in December, but it's one that you've studied at least to some extent before. So moving chromosomes around inside the cell is an important function. But other functions that you might not be as familiar with are maintaining what's called an asymmetric cell shape, which is demonstrated in the first two examples up there. So if you take the microtubules away from most cells, and there are drugs that will do that, then the cell just kind of, becomes this little circle. Okay? If you want any kind of asymmetric shape, then microtubules are really going to help you along with that. And probably the best example of that is shown here in the nerve cell. Right? So nerve cells are not just little spherical cells, right? They tend to have a cell body, and then a really, really long axon, and then a terminal knob. Well, what's maintaining that really, really long axon? The microtubules, as it turns out. So centrosome is up in the cell body, and all these microtubules run down the length of this axon, so all the plus ends are down here. And it is clearly the microtubules that maintain that axonal process. And also the little dendrite, the little tiny extensions that usually come off the cell body are also maintained through microtubules. How do we know that? Well, because if we throw the drug that causes all the microtubules to fall apart at a nerve cell, guess what? The axon goes, all the dendrites go, and we get a circular little nerve cell. Okay, so those experiments are quite easy to do. Okay, so we can really often tell if a microtubule has something to do with a specific thing in a cell. We can show that by taking all the microtubules away and seeing if it has that effect. So that's a really good example of maintaining an asymmetric cell shape due to microtubules. Kind of on a smaller scale, as shown here, this specific example here is what's called a ciliated epithelial cell. So these are the types of cells that line your airway, line your trachea. And the little extensions at the top, the little cilia, are maintained by microtubules. And we'll take a more close look at cilia and flagella in a little bit. But just in terms of general function, so those little projections are maintained through microtubules. Again, how do we know? Because if you take a ciliated epithelial cell and you throw the drug at it that depolymerizes the microtubules, guess what happens to the cilia? They shrink, they go away. Okay, so it's very clear that they're maintained by microtubules. So asymmetric cell shapes. Another good function of microtubules, and a really good example of it is in red blood cells, is what I usually call sort of like helping to maintain sort of the structural integrity of the cell. So in red blood cells, think about what we put those poor cells through all the time as they go from larger arteries to smaller veins to really tiny capillaries. Right? They're put under an enormous amount of pressure. Right? And if you put cells under too much pressure, what tends to happen to things? Think about a water balloon. If you put too much pressure on it, what happens? It pops. Well, you put cells under too much pressure, they're going to pop too. That would be bad, right? You don't want your red blood cells popping open all the time. So to help that from happening, there is a ring of microtubules, among other proteins, but there are microtubules involved, that run around the circumference of the little red blood cell. That's called the marginal band that you see here, a marginal bundle. So it runs all the way around, and it kind of gives structural support to the cell membrane especially, as these cells are put under that kind of tremendous pressure, especially as they're running through the capillaries. So it helps the cells from basically popping open under that pressure. So there are a lot of good functions for these microtubules, and we'll come across some others as well. I might as well mention it here, I guess. Oh, well, here's the next one. Okay, so before we move on, do these functions here make sense? Okay, so more on the cell division stuff later, but that's certainly an important one. We're okay with asymmetric cell shape and structural integrity kind of ideas? Okay. Yet another function is that in most cell types, probably all cell types, the microtubules act as sort of a highway system in the cell. So there's stuff, and we've already sort of looked at this already. Think about all those vesicles that we were moving from the ER to the Golgi, and then the Golgi to the cell membrane, that kind of stuff. They're all moving, actually, as it turns out, along this sort of microtubule highway. So they're not just floating around randomly, as maybe we once thought. They're actually being moved along microtubule highways. And they're being moved by these, what are called motor proteins. And there are two big families of motor proteins. One is the dynein family, and the other is the kinesin family. And remember the polarity idea, right? We have the plus end and the minus end, and that's how the two families are going to differ. The dynein family moves stuff towards the minus end, whereas the kinesin family moves stuff towards the plus end. So they can each only walk along microtubules in one direction. Okay. And they're a family of proteins because, while they all share that characteristic, so all kinesins walk towards the plus end, but they differ in terms of what they're moving. Okay, so one type of kinesin moves, say, I don't know, vesicles. Another one moves mitochondria. Another one moves, if we're in plant cell, chloroplasts. So they differ by what cargo they carry or move, but if you're a dynein, you're always moving towards the minus end. If you're a kinesin, you're always moving whatever your cargo is towards the plus end. Okay, so that's kind of the general idea. So again, all the kinesins walking various cargoes towards the plus end, all the dyneins walking towards the minus end. So again, this polarity idea really, really important to these microtubules. And this walking along these microtubules, as you might imagine, costs energy. Right? So basically, if we kind of think about these two little extensions here as sort of feet, every time the dynein or the kinesin takes a step, it costs you an ATP. So it's sort of a three-step process that I will try to badly demonstrate here. So the ATP binds, and one of the feet goes up. The ATP is hydrolyzed, and it's planted. The ADP is pushed off, and the back foot is sort of dragged up. ATP binds, hydrolyzed, kicked off. Binds, hydrolyzed, kicked off. So every step costs you an ATP. And as you might imagine, it's going to take you a lot of steps to walk along a really long microtubule. So let's link this back to that little piece of trivia that we learned way back in metabolism, that each cell is using like 10 million ATPs every second. Well, here are some things that they're doing, right? They're moving cargo all over the place inside the cell. Every time any one of these proteins takes a step, it costs you an ATP. So here's an awful lot of ATP being used just to move cargo around the cell. So once you start thinking about all the things the cell is doing all the time, you can probably come up with 10 million ATPs pretty simply. Patrick?
But it costs less ATP to go towards the minus end.
Well, that's an interesting question. I don't think so. Although I don't know that for sure, but I don't think so. So you're thinking they're just sort of riding along that. I don't think so. I think that might play a little bit of a part, but I think that's still costing you an ATP every step. But that's an interesting idea. Yeah, but my gut reaction says no. But I don't know that for 100%. Okay, so again, here's sort of that model. Okay, that sort of three-stage model there. So it kind of makes some sense in terms of how that's working. Does that idea kind of make sense? Moving stuff around? So this seems to be a very, very vital function of microtubules. And again, how do we know? Because if we take all the microtubules away, then everything just sort of starts randomly floating around the cell. And that ultimately might be okay, but if we think of something like, say, this nerve cell, yeah. Okay. Some axons are like 1 meter long. So if you make a vesicle, say, filled with neurotransmitters up here, which is where they're made, but they need to get all the way down here to the knob where they're going to get released, and this axon is like 1 meter long. If they were just randomly floating around the cytoplasm, some people have estimated it would take weeks for it to just randomly diffuse down the length of that axon, because sometimes it would go this way, sometimes it would go that way. You know, maybe eventually it might get down here, but it would take a tremendously long amount of time. But if we just hook that vesicle up to, let's see, we're going to move it to the plus end, so we're going to need a kinesin. I always forget that. Yes, a kinesin. Then all it does is walk towards plus ends. So if you hook it up with one of those motor proteins, then you're sort of saying, make sure this gets all the way down to that end of the cell. So it's going to take a much, much shorter time. It's going to cost you some energy, but that balance, that payoff is sort of worth it, because otherwise, you know, most of your vesicles filled with neurotransmitters are not going to be where they need to be. And so especially in that kind of extreme example is a good one, though, in terms of why this would have developed, why would this have evolved. Well, it's important to get the cargo to its destination in a reasonable amount of time. You know, you can make the easy analogy to the FedEx or the Postal Service, right? You don't want it just kind of randomly floating around the world and eventually it gets to your house. You want to get it to your house as quickly as possible, right? It's sort of the same idea. Okay, does that kind of make sense? Using these microtubules as sort of a highway system or a transport system. Okay, and again, we do have microscopic evidence of this. So here's some kind of little vesicle, and the proteins attached to it kind of are walking it along this microtubule here. So again, just to link this back to what we were talking about earlier this week and late last week, all those vesicles that are moving from the ER to the Golgi to the cell membrane, and sometimes moving in from the cell membrane or backwards to the ER, all those vesicles are actually being moved in this way. So they're not just randomly diffusing over to the Golgi. They're being walked along microtubules to the Golgi. And they're being walked from the Golgi to the cell membrane. So this links nicely to the stuff we were just doing. Okay, so far so good? Okay. The next thing we want to look at is, again, another of the main functions of a microtubule is to be part of the structure, we've already seen of cilia, but also of flagella. And as you're probably already aware, flagella are structures that allow certain kinds of cells to move. Perfect example are sperm cells. Right? They have that long flagellum, and as we'll see, it's going to sort of wiggle back and forth and allow that cell to move. Okay? So we're going to kind of take a look at that aspect of microtubules as well. Well, this brings us first to a second kind of organizing center. And it's the one I mentioned earlier called the basal body. And as we'll see in just a moment, the basal body really looks surprisingly similar to a centriole. So here is a cross-section through a basal body. And we start at the top, 1, 2, 3, 4, 5, 6, 7, 8, 9 triplets of microtubules. So it looks surprisingly like a centriole that was embedded in that centrosome in animal cells. But it's a little bit different in that they're not 3 complete microtubules. So in centrioles, it appears that you have 3 complete microtubules that are just stuck together. In a basal body, it's more like that, where you have 1 complete one, and then the next 2, so these are usually lettered, A, B, and C. So the A one is complete. It has 13 microtubules, or 13 protofilaments. The B and the C, though, are only partial. They only have 11 of their own, and then they sort of share the 2 protofilaments with A, and C shares 2 protofilaments with B. So there's a little bit of a different arrangement of the triples, but they're still triplets, microtubules. And there are other proteins that are, of course, holding these things together. And the other big difference, the centrioles, of course, as we saw, there are always 2 of them that are at right angles. That's not the case for a basal body. It's just one of these things. There aren't 2 of them stuck together or anything. But there are some striking similarities between a centriole and a basal body. And so this is what's at the base of a flagellum or a cilium. And so if we picture a sperm cell, so we have the head region where all the DNA is, inside the nucleus. We have what's called the neck region, where it's filled with mitochondria for energy, and then you have the flagellum itself. Well, really all the flagellum is a long, as we'll see, a long set of microtubules that push the cell membrane out really, really long. It's really, really long and thin. So this is covered by cell membrane with microtubules sort of pushing that out. So the basal body would be basically right here at the tip, or not the tip, but the starting point of the, in this case, the flagellum, or if we're back in those ciliated epithelial cells, at the base of each one of those. So actually, it might be worth going back to that picture. There it is. Okay, so in this little version here, there are 4 cilia. There are 4 basal bodies then. So at the base of each one of these cilia is a basal body. So another good example of that, if you've ever, like in high school, seen a paramecium, right? The little protist that has like hundreds or thousands of cilia. Each one of them, at the base of each one of those cilia, is a basal body. Okay, so same thing in our ciliated epithelia, same thing here for this sperm flagellum. And so at the base is a basal body, and that somehow organizes the microtubules that make up the rest of the structure. Exactly how any of that is done is still really not well understood. But we do know that it's an absolute requirement. If I was good enough with a laser and I zapped that basal body out of existence, that cilium would, all the microtubules would depolymerize and it would cease to exist. So it is an absolute requirement. We just don't know exactly how that organization happens. But it is an absolute requirement. Okay, so that's at the base. And then as you move out into this flagellum itself, the arrangement of microtubules changes. It changes from triplets that we see in the basal body to doublets, as you see here, but still 9 of them. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so the 9 triplets become 9 doublets. So 9 around the circumference. And then, and this has no parallel in the basal body, right? The center is sort of open. But then running up the length of, up the middle, basically, of the flagellum or the cilium are 2 complete microtubules. So this is usually referred to as the 9 plus 2 arrangement. 9 doublets around and 2 running up the center of the structure. This has the 9 plus 2 arrangement. And then there are all kinds of other proteins. As you can see, you don't need to worry about them, but they put some of the names on here. But you can see all these other proteins that are cross-linking all this stuff together and making kind of a complete structure. The one, though, that we do need to think about is dynein. Oh, we just learned about dynein, right? What's dynein? Yeah, one of those motor proteins. Which way does dynein like to move? Towards the minus end. Okay, so keep that ready in just a second. Because now we want to figure out how all of this creates motion. So, again, if you kind of take a cross-section through the flagellum itself, that's what you see. Again, the 9 doublets around the circumference, the 2 complete ones going right at the center, all of these other proteins linking all this stuff together. Dynein is going to be, for our purposes, sort of the most important, because that's what's going to create the motion. So this is just showing you kind of cross-sections at various levels. Okay, so here's the basal body, so our 9 doublets. I'm sorry, 9 triplets. Then as the flagellum itself starts, you have the 9 doublets. And then if you go up a little bit further, then the central ones sort of appear. So again, we do have microscopic evidence for all of this stuff. Here we go. Okay, so how does that structure then create motion? Okay, so cilia, sort of, to keep it simple, sort of have kind of like a windshield wiper kind of motion. They kind of go back and forth this way. And they can either propel the cell, so if you're a paramecium, then these things are moving back and forth and the whole cell is moving. If you're those ciliated epithelial cells of our trachea, those things are moving back and forth, and they're moving mucus out of your lungs and up to your throat so you swallow it. Okay, so the mucus traps all the dust and debris and bacteria and stuff that we're breathing in all the time. So it gets stuck in the mucus. These things beat upwards. The mucus moves up out of your lungs, and you swallow it and get rid of it. And we're all doing that all day. If you think that's gross, we're all doing it all day, every day, quite unconsciously. Okay. But that's the main system that keeps your lungs, from a microbiological standpoint, sterile. Okay? And that's part of the problem with cystic fibrosis. Remember we talked about that a little bit ago. That was a problem with the transporter, right? A chloride transporter, right? It makes the mucus too sticky, and your poor cilia can't really move it very well. So it's even more sticky, so it's trapping even more stuff, but then you can't get it out of your lungs very well. So that's what most CF patients die of, is just massive pneumonia, because they can't get the bacteria out of the lungs. Okay? So they're kind of moving back and forth, kind of like a windshield wiper. And then the longer flagella, like on this protist, like on this euglena, or like on a sperm cell, sort of goes, if you're an Indiana Jones fan, and if you look at a whip moving down the length, that's kind of how these work. So it kind of wiggles back and forth, kind of whip-like, and that creates motion, basically. So eukaryotic flagella, like we're looking at, work very differently from bacteria. So bacteria can have them as well, but the bacterial kind spin 360 degrees, much more like a boat propeller. Eukaryotic ones sort of wiggle back and forth, much more like a whip-like motion. And we're out of time, aren't we? Okay, so here's our cliffhanger for the weekend. Okay, we'll need to figure out how all this comes together to make motion. Okay, so